Good afternoon, everybody. My name is Kim Hellmans, and I'm going to be moderating this conversation, the Science Cafe, on the topic of vaccines against COVID-19. So where are we so far? And I have the great pleasure of introducing our speaker today. So I'll do a brief introduction, and then Dr. Rudd will come on and give his presentation, and then we'll have some time at the end for a moderated portion of question and answer. So in brief, Dr. Erling Rudd is a scientist in residence and adjunct professor in the Department of Health Sciences at Carleton University. He has been a research scientist for 31 years following his PhD, and he has much experience in academic, biotech, and pharmaceutical, as well as government sectors across both Canada and the US, as well as the UK. And this has been in his capacity as a virologist. He has over 40 years of postgraduate experience working in many aspects of virology, and 38 of those years has been on emerging infectious diseases. So he's an absolute expert in this topic, and we are very, very lucky to have him speak to us today on the topic of COVID-19. This is because most of his experience has been with working with infectious diseases that are viral. And these are from RNA viruses, such as California encephalitis, as well as retroviruses, such as HIV. And his activities have ranged from both basic and molecular virology to antiviral and vaccine research and development. While he's worked mainly in the private sector for the Wellcome Foundation, for example, as part of GlaxoSmithKline, and within the federal government of Canada, with Health Canada and the Public Health Agency of Canada, or what's called PHAC. He has published over 55 peer-reviewed publications and has presented over 165 invited talks about his work at conferences and universities around the world. So please join me in welcoming Dr. Rudd. It's unfortunate that we can't have this big space and be all in one physical environment, but due to the restrictions, and we are following good public health advice, and we're doing this in the virtual world, which will actually probably allow folks that may not normally be able to come physically on campus or in other spaces to be with us today. So without further ado, I'm going to turn off my camera, I'm going to turn my mic off, and Dr. Rudd should appear on the screen momentarily and start his talk.
There we are. There we go. Okay. Good afternoon. I guess it's about 1:35 now. So I have about 30 slides to go over. But before I actually get into my talk, I thought I'd mention something personal. I am married. I have 2 children. One daughter actually was just married in Nova Scotia on the 5th of September, which required that I go to Nova Scotia and spend 2 weeks in isolation or self-isolation with my wife. Fortunately, at my uncle's house, which had a nice backyard, so we actually could get out and take a look at the deer and other things. So it was quite nice, actually. The wedding was lovely, beautiful weather, etc. So feel quite fortunate. It actually was quite nice to be in Nova Scotia. So I'll get into my slides. So today's topic is very much vaccines against COVID-19, or, if we want to call the virus, it's called SARS-CoV-2. So SARS coronavirus-2 versus the 2003 SARS, which is now called SARS-CoV-1. My name is Erling Rudd. Yes, I'm at Carleton University. I'm also an adjunct professor over at the University of Ottawa in pathology and lab medicine, just because I don't have enough to do. Next slide. So worldwide, as of October 10th, and this is information from Worldometer, there were over 37 million coronavirus cases documented, over 1 million deaths so far, and 27 million recovered. That's amazing considering we only first recognized this virus in December 2019. So to have that number of deaths is quite amazing within a year for a brand new virus. In Canada, Well, we've had 180,000 new cases. This is some information from CTV News. Actually, they have a tracker tracking every case every day, etc. And this is sort of the October 10th version. As you can see, there was just under 10,000 deaths in Canada to October 10th. If you look at the panel underneath here, you can see that our cases, or the 7-day average cases, have been increasing exponentially pretty much since September. So we are in a second phase, second wave, what do you want to call it? So what public health measures can we use to slow down SARS-CoV-2, which caused coronavirus or COVID-19? Well, we've been told to physical distance, keep 2 meters between us. Frequent hand washing, as you know, soap and water is great. If you have to use ethanol products, use them. Over 70% is the best. Proper cough etiquette. We don't hear about that much anymore, but at home I try to make sure that I do cough into my elbow just to prevent spraying droplets and aerosols around the house. And lastly, wearing masks to slow down the spread of the virus. This was quite controversial, still is in certain parts of the world, but there's no better way to actually limit the transmission than to wear a mask to prevent the droplets from spreading as much as possible. What is our goal? Well, the goal would to develop a safe and effective vaccine for COVID-19. And it may actually be our only hope of stopping this pandemic. There's lots of discussion of, just let it go, like Sweden. Let the numbers of infected people increase to 80, 90%. Well, that's basically been described as unethical by the WHO and others. How long will it take to develop a vaccine? Well, I indicated in my sort of description that it's 12, 18 to 24 months. And I still state that even at this point, we're probably, to get it into everyone's arms, 12, 18 to 24 months away. So how long does it take? I don't know if this is showing up well. I'll try to move this. There we go. To develop vaccines. Well, typically, varicella took about 28 years to develop a chickenpox vaccine. Flu mist, 28 years as well. Human papillomavirus, 15 years. Rotavirus, 15 years. Now we're trying to develop a COVID-19 vaccine in 18 months. That's amazing, absolutely amazing. If we can do it, hats off to everyone involved. Well, let's describe a little bit about the product development pipeline. Why does it take 18 months? Why does it take 15 years or longer? Well, if you look at every aspect of how you would develop a product, and this isn't unique to vaccines, you use this for antivirals, for any other drug. You look at a requirement setting to identify what your needs are, do basic research to basically find out, in the case of this, what the virus is. Are there any immune responses that may correlate to protection? Applied research goes right through the process, right to clinical trials. Then preclinical development. Once you have an idea, you've generated something you think will be a vaccine, you try to do animal studies. They're called preclinical safety and efficacy studies. Usually the number of animals you have are not going to give you something that's highly statistically relevant, but it'll give you an idea of maybe this is working. The next process is process engineering and small scale production. And this is really a difficult phase. Universities will have a lot of great ideas, but to actually get process engineers to take that idea into a product, a small scale production, is not trivial at all. Then once we do that, companies and others will apply to FDA and Health Canada and others to do phase 1 and 2 safety and immunogenicity clinical trials. These are extremely important. It's usually in small numbers of people, 50 to 100 people. Just to see, is this vaccine actually inducing anything, and is it safety, or is there some problems right at the get-go, which would stop this product development? Then you go into phase 2B and 3, which are efficacy trials, and that's pretty much where a lot of the vaccines will end up. And if there is actually some indication of efficacy and they're safe products, then they'll take it further for regulatory approval and licensure. And once that happens, there's large-scale production and manufacturing of lot releases that have to be tested to ensure adequate supplies, etc. The provinces get involved, federal government gets involved in actually acquisition of these vaccines. And then ultimately, once the vaccines are brought into the general community, there's post-market surveillance for any adverse events. Health Canada keeps in a tab on what's going on that. So how's Canada doing? This is from when I worked at the Public Health Agency. We actually were working on how we could stimulate new vaccines. This is before COVID actually showed up. So if you look at the maple leaves underneath it, it's a fast way to look at it. Where are Canada's strengths? Well, we're great on requirement setting and basic science and applied research, a little bit less, but pretty good. Preclinical testing, we have some real good capacity. Our biggest issues are scale up. We were trying to do a study right at this point. We're trying to develop, but we didn't have it. Underneath that, you'll actually see the valley of death indication. And this is really where the product will actually live or die. Okay, so if it doesn't get through this, the product dies. That's all there is to it. So we definitely call it the valley of death. Then we have greater capacity, definitely on licensure. Health Canada is very involved. Production, manufacturing, a little less in this country. A lot of that's done overseas in the United States. Purchasing, stockpiling, Health Canada, Public Health Agency are very involved in that, and post-market surveillance likewise. So COVID vaccines, where are we with that? Well, if you actually take a look at the amount of investment, it's amazing. These are billions of dollars. So you can see that BARDA, this is in the United States, and Operation Warp Speed out of the US, has already spent probably just over 11 million or $11 billion to try to develop a vaccine. The EU Alliance, just over 4 million, or almost 5 million, or 5 billion, sorry. The UK government, just over 4 billion. Bill and Melinda Gates Foundation, just over 2 billion. CEPI, that's the Coalition for Epidemic Preparedness Innovations out of Oslo, Norway, has actually invested over 2 billion, or sorry, over just about $1 billion in that. And the government of Brazil likewise has invested tremendously. There's obviously a lot of other investments, but that gives you an idea of what the investment is like within a year. To develop a vaccine, you're usually talking about $1 billion to develop a vaccine. So this amount of investment shows that there is a major effort. Where are phase 3? Now, when I was talking about the different types of clinical trials, phase 3 are what you want to get through. They're proving the efficacy of that vaccine. Is it safe? Is it effective? You're going to test that. The only place you can really test those quickly would be in high prevalence populations. That is in areas where people are being infected in large numbers. So the United States, for example, Brazil are 2 examples of that right now. There's many others, obviously, you can see from this. Each one of those points is where some phase 3 clinical trials are going on in the world. So you can see it is truly an international effort. People are working together tremendously to try to get a vaccine as soon as we can. So I took this actually from the Financial Times. They produced a really nice review of where we are. And if you actually take a look at it, this is sort of a setup of a phase 3 clinical trial where they're trying to get 30 to 60,000 volunteers involved. Half of them would be given the vaccine candidate, and the other half would be given some placebo. Okay. Now, if you look at the panels underneath, it's sort of a breakout. If you have a vaccine that's only 10 to 20% effective, if you look at the vaccine group versus the control group, you don't see a large number of differences. However, if you have something like the measles vaccine, which is probably about 97% effective, you see a tremendous difference between the control group and the vaccinee group. That is what you're looking for. Now, usually when we're actually, what they're trying to do is event-based trials. So they'll halt their phase 3 trials when the 2 companies involved here would be Moderna and Synvac, have 151 to 154 symptomatic infections in these 2 groups. Okay, so they are in the middle of doing that. I understand that they should be breaking out the results sometime in November. The FDA is trying to get, the Food and Drug Administration in the United States, trying to get 50% fewer symptoms as something that they would consider for licensure and to go forward. So we're looking for something like that. So who is building the biggest vaccine portfolios? That is, who is sort of already shelling out money to try to get vaccines for their populations? If you actually take a look at the top 5 here, the UK, US, Canada, Japan, the EU, and Australia, they basically are trying to get a coverage of at least 3 doses to, 3 doses to over 5 doses per individual in their country. That'd be per capita rates, which is amazing. But other parts of the world obviously either don't have the resources or haven't committed the resources to this point. India, Indonesia, Brazil, for example, Switzerland and China haven't put as much investment, at least to this time point. That was the Financial Times paper in early October. So there has been a really nice review actually put together by Emily Chung from the CBC News, where she was looking at COVID vaccines as well. And in her October 6th paper, she actually saw 151 preclinical evaluations of different vaccines. It's amazing, 151. 19 phase 1, 13 phase 2, 10 phase 3. So phase 3 are really the ones that are getting close to the point where they could be licensed. The one approved here is the Russian product called Sputnik 5. An amazing name. I believe the Russians decided to approve that product before their phase clinical trials were completed. So it'll be interesting to see how their vaccine actually ends up doing. Now, vaccines will come through this pipeline. Some of them will work out well, some of them won't. So whatever's got through the phase 3 clinical trials may not be our best bets in the end. We may have to wait for better products, but I have a feeling that some countries are going to go ahead with whatever they get. So what are the types of vaccines out there? There's a variety of them, from the traditional live attenuated and inactivated virus vaccines. These are the vaccines that traditionally have been used for most of our vaccines, okay? Whether it's flu vaccines, polio vaccines. The first polio vaccines were live attenuated and inactivated vaccines. These were whole-killed vaccines. Work very effectively. We're eliminating polio from the world. The next ones are non-replicating viral vectors. So they actually have used viruses, made them non-infectious. That is, that they'll go in and infect a single cell, but they won't replicate, and use that as a way of bringing a gene from a vaccine of interest, or a virus of interest. In this case, for example, the spike proteins gene would be inserted into the viral vector and used to express spike protein and induce an immune response. Another approach is using a sort of a live replicating viral vector. If you think of vesicular stomatitis virus, some of the virus vaccines are actually live attenuated, and that is that the virus will actually replicate to a certain level. The two viral vaccine or other approaches that are being tried right now, there's a lot of activity, are the RNA and DNA vaccines. There's been a lot of activity in those areas. They're very rapid at setup, but to this point, there hasn't been a lot of approved vaccines using those technologies. Peptide vaccines are out there. I won't talk about them. There isn't a lot of activity, I don't think, in COVID at this point. Virus-like particles, lots of activity there. The advantage of those is that you're going to induce better immune responses because you're having multiple copies of the protein of interest expressed, inducing better immune responses in the individual. And recombinant proteins is another approach. That's using the recombinant DNA technologies to express proteins within a variety of expression vectors and purify them to a high level. The unfortunate thing about those is that they take longer to produce, and in some cases they require adjuvants. How do actually vaccines induce their protection? I'm not going to go through this completely, other than to say you inject the vaccine and you'll either induce an activation of B cells. These are the cells that produce antibodies. They'll mature into what they call plasma B cells, which are expressed as antibodies. Some of those antibodies will actually bind to the virus of interest and may neutralize it or not. So we're not absolutely sure that all the antibodies we may be inducing with some of these vaccines actually will be neutralizing. There may be other types of approaches, but that's not the only part of the immune system. There's also the T cell response. Okay, so you might have heard of natural killer cells. Well, they actually will take the antigen presenting cells, their activated T cells, and actually induce a natural killer cell to identify virus infected cells and kill them. Okay. What you're trying to hope in all cases is induction of a memory response. And the memory response will allow you to respond if you come in contact with this virus in the future. So here's a list that I got from the London School of Hygiene and Tropical Medicine. Currently, if you take a look at the number of vaccines out there, just looking at RNA vaccines that are in preclinical, they have 25 RNA vaccines. 2 that are in phase 1, 2 that are in phase 1/2, 1 that's in phase 2, and 2 that are in phase 3. These were the first vaccines that actually came through very rapidly after this virus was sequenced in January. Moderna, one of the companies, and one of the other companies were very rapid off the block and got their vaccines going. DNA vaccines, likewise, a little fewer, 2014. They're very much at the phase 1, phase 2 levels. They haven't got to phase 3 at this point. Non-replicating viral vectors, there are 4 that are in phase 3 clinical trials. The others are fairly early in the product development pipeline. Replicating viral vectors, there's none that have actually made it to phase 3 clinical trials at this point. Inactivated viral vaccines, okay, so these are the traditional approaches. There's 7 that are in preclinical testing, 3 that are actually in phase 3 clinical trials. I believe most of those are out of China. Live attenuated, there's 4 in preclinical testing or development. None of them are in phase 1 to phase 3 clinical trials at this point. Protein subunits, a lot more activity, 62. 8 that are in phase 1, 2, 1 that's actually made it to phase 3 clinical trials. And virus-like particles, an interesting approach, and actually there's a Canadian initiative on this. There is 13 in preclinical development, 1 in phase 1 clinical trial, and 1 in phase 1/2 clinical trial. And others, I don't really know what they are. There's 32. The people involved actually haven't disclosed exactly what they're doing, but there we go. So imagine that all this activity within 10 months. It just blows my mind that there's actually that number of vaccine candidates in the pipeline at this time point. So let's go through the different ones and see what's out there. What is the advantage and disadvantage of RNA vaccines? Well, the RNA vaccines are fairly easy to produce and scale up, but and no virus is needed to make the vaccine. So you can take the sequence, you can generate a product without actually having to isolate the virus. But you don't always produce a good immune response compared to whole viral vectors, and may require some adjuvants to induce better immune responses. We're still sort of wondering about what will happen there. You can see there's 2 products that are in phase 3 clinical trials, the Moderna product and the Pfizer product. CureVac and Arcturus at Duke University are in phase 2 clinical trials, and Imperial and the People's Liberation Army, Academy of Military Science, are in phase 1 clinical trials. So this is really new and novel. It's going to be very interesting to see what happens with these results. For the most part, the Moderna product, very innovative. They're looking at something, they're trying to deal with the fact that sometimes you'll induce antibody responses that may actually not be neutralizing and may actually bind to the virus, but may not neutralize it. The next group is the DNA virus. It's very similar to RNA. It's quick and inexpensive to manufacture in large quantities. The shelf stability is not a problem, really. You don't require freezing of this. So if you want to get this into anywhere in the world, it's fairly easy. You don't need to have freezers. It's easy to switch to different gene products. Once you have your system set up, you just put another gene into it, bang, away you go. The problem is that you usually require some form of adjuvant to get good immune responses. And this has been a thing that sort of slowed the DNA vaccines down. I remember seeing some of the first presentations back in about 1990, and we're still trying to get the DNA vaccines going. There are 2 Canadian initiatives, Mediphage Bioceuticals from and University of Waterloo has one, and Entos Pharmaceutical from Edmonton, I believe, is another product that is in preclinical evaluations at this point. And you can see there's, 4 others that are in phase 2 clinical trials using this approach. Non-replicating viral vectors, you get a more powerful immune response than subunit proteins, that is taking the protein itself and purifying it and just injecting. And some of them actually don't need to be frozen away, so that they can be used in resource limited tropical areas. The downside is people have already been exposed to some of these vectors. For example, the adenovirus. We've all come in contact with adenovirus. Ad5, we've pretty well all seen it. So if you already have an antibody response to the vector, you may recognize that vector and never actually get an induction of the protein that you're interested in vaccinating against. So it could be a bit of a problem. It's harder to scale up than proteins or DNA because the virus still needs to be grown up on tissue culture or other systems. Because each virus can only infect one cell, large quantities of the virus need to be produced. You have to produce amazing amounts of this virus. So though it's very interesting, it still requires a lot of work. And you see examples of this. The adenovirus MVA, which is modified vaccinia ankara, a weakened pox virus, parainfluenza, and rabies. Examples of this are wide, and for example, the AstraZeneca product that actually is using a chimpanzee adenovirus. Very interesting. The phase 2 clinical trial has been stopped because there were some problems, but that's not irregular. When you actually are giving a vaccine to 30,000 or more people, in that population, there may be people that have pre-existing problems that just seem to surface at that point, but they have to investigate what the problem is and see if it is associated with the vaccine itself. So that trial was stopped and then restarted again everywhere except in the United States. Ganlia Research Institute, that's the Russian institute that has approved their product called Sputnik 5, which is an adenovirus product. Johnson & Johnson has a phase 3 clinical trial again, and I believe they just stopped their phase 3 clinical trial again because of no adverse event. There's other companies out there as well. They're at different earlier phases of clinical trials. Replicating viral vectors. These are similar to the non-replicating, but they'll induce fairly strong immune responses because you're infecting, an infection occurs, the immune system is activated, and you should get a fairly good immune response to the gene you've inserted. Vesicular stomatitis is an example of this, and that was used as the vector for the Ebola vaccine that the group in Winnipeg actually generated, and it was tested in West Africa in 2016. The downside of this is that more testing is required before approval than protein or nucleic acid-based approaches. They need to be stored, transported, and cooled, so you actually need to have that cold cycle dealt with, and that can be a problem for certain parts of the world. Examples of that are fairly early on, but you can see the University of Manitoba and Western University in London are both involved in preclinical testing of some of these approaches. I believe they're VSV approaches. The inactivated virus approach. Well, this is a traditional vaccine approach. Unlike live virus vaccines, it actually can give them to people with weakened immune responses. So if there are individuals that have, for example, HIV, their immune response is not that great, you can actually still give the vaccine to them with this approach. The downside is that because it's killed, it doesn't necessarily induce a very good immune response as compared to live viral vectors or live attenuated vaccines. Several doses can be required, so you may see an initial vaccination and then boosting required. And it also may require adjuvants. Okay. Examples of this are all over the place. Sinopharma in China and Sinovac are both in phase 3 clinical trials. The Wuhan Institute of Biological Products, or Sinopharm, is also there. And there's other products through, I believe, India and China that are at different levels of clinical trials. The live attenuated virus approach, again, this is something I worked on in regards to HIV. Very interesting approach because you actually are using the virus itself or an attenuated version of the virus. You can give it once, you don't have to boost with anything else. So you're giving one dose and away you go. However, it may not be suitable for people with weakened immune responses. When we actually took a look at one of these approaches using a monkey model that I was studying, we actually saw in very young individuals, AIDS-like symptoms were being induced, even though it seemed to be fully safe in adults. Live virus need to be refrigerated, otherwise they'll die. And the virus must be grown to large quantities, again, which means scale up is a bit of an issue, and it costs a lot of money. There's really none that I know of that are in clinical trial at this point. Protein subunits, well, they can be produced fairly quickly. Since the development of recombinant DNA approaches, we've been expressing proteins. There are a variety of methods, either using bacteria, using yeast. One of the first recombinant approaches that was used was for hepatitis B, and it was generated actually in yeast. Can be produced fairly quickly. However, it doesn't generate as strong an immune response as whole virus vaccines or live viral vaccines. A compound called an adjuvant is needed, so adjuvants are needed to boost that immune responses. And they can't be scaled up as quickly as RNA or DNA. However, they're fairly rapid. As you can see, there's lots of candidates, and there are some Canadian candidates. Vito Intervac from the University of Saskatchewan is working on a product. University of Alberta, likewise, and IMV Inc. is working on a product as well. But there's a lot of other product, and you can see Novavax is in phase 3 clinical trials with their protein product. Virus-like particles, very interesting approach. They actually would generate, like I mentioned, the hepatitis B product was a virus-like particle, actually. That will induce very good, strong immune responses. I think I have an unstable internet connection. Okay. It can be produced fairly quick. The cons are ensuring the stability and purification in production time, and can be hard to produce in large quantities. Examples of this, in Canada, actually, we have a product called from Medicago. They actually generate their product in plants. Very interesting. They're in phase 1 clinical trials, and University of Manitoba has another product that's still at the preclinical level. So there's lots of Canadian candidates, but if we actually look at where Canada's invested to this point on buying potential vaccines, we haven't bought a Canadian product yet because there's none actually that have made it through to phase 3 clinical trials at this point. However, because there's a big demand for these vaccines, we may actually need a Canadian vaccine. And as Volker Kurtz has been quoted here as saying, I don't think we want to rely on others, hoping that they will remember us when the vaccine may be limited. So there's a big question mark there. So is the Canadian products, as you can see, Medicago is in phase 1 clinical trial. They have their phase 1, their virus-like particle. There's a DNA approach from University of Waterloo. University of Manitoba has virus-like particles as well, Vito Intervac, protein subunits from the University of Alberta, replicating viral vector from the University of Manitoba. Western has a replicating viral vector. Entos is DNA, and IMV is a protein. So we have a fairly large sort of product development pipeline with different approaches, which is great because you don't want to actually put all your eggs in one basket. So in summary, and I think I've talked about half an hour now, it's just amazing the amount of vaccine R&D that's actually gone on within a year. Absolutely amazing as far as I'm concerned. There's at least 10 candidates in phase 3 clinical trials. And those are really phase 3 clinical trials that are being tested in areas of the world that have high prevalence populations. That is where lots of people are being infected. So you can actually look at how many people are being infected in your vaccine group versus how many people are being infected in your control group or the placebo group. However, with all this good news, we have to remember the vaccine development, as I mentioned in one of my first slides, can take up to decades to develop. We've been working on HIV vaccines for 30 years, And we still don't have one that is truly effective. We have things that are partially effective, but we don't have something that is, for example, 70% effective. Other vaccines, however, have been brought out faster. So the Ebola virus vaccine has definitely been brought out faster, and it was tested in Africa. Great work. But we have to remember, safety and efficacy have to be the gatekeepers. We have to make sure that the products that are being tested go through those phase 3 clinical trials and are determined as being effective and safe. Will we actually be able to get vaccine coverage of more than 70%? Well, I've heard stats that people are sort of less prone to take the first vaccines than they might have been, say, 3 or 4 months ago. But we have to look at vaccines may be the only thing that will actually induce herd immunity here. There's some recent things, actually, was in today's newspaper, that showed that there has been infections in individuals that were previously infected. They seem to have been reinfected fairly quickly. So it looks like if there's a protective immune response that's being induced in people that have been infected and have recovered, it doesn't seem to last very long in some people. How long will the immunity last, even with vaccines where we can induce maybe better levels of antibodies and better protection? Is it months, is it years or longer? We still don't know. We're not there yet. How long will it take to roll out a vaccine to billions of people? 12 to 24 months before it would be widely available is sort of my prediction at this point, but it all depends on how well these vaccines actually get through the phase 3 clinical trials and can be truly scaled up to those levels. COVID is an absolute global problem, and it needs global solutions. And I think from what I showed you today, we have a large group of people from across the world that are working on COVID vaccines, and I really take my hat off to everyone who's working on this. They're working extra hours, without a doubt, long hours, to try to get something that's going to work and help us all out. However, what can we do? Well, because they're responding, we should also respond by making sure that we continue to wash our hands, wear masks, keep at least 2 meters from others in public spaces when possible, and use cough etiquette. And I think that's it for my talk.
Thank you so much, Dr. Rudd. We have a number of great questions in the chat, so I'll start. One of them, I think you might have addressed, but somebody asked, most of the Canadian products seem to be lagging behind in trials. Why is this?
Well, I think when I showed you how we were doing in Canada, and in that one slide, I won't show it again, but with the maple leaves, I think if you actually take a look at our clinical trial capacity, we don't have as much clinical trial capacity as we could have. Partially it's just the number of people we have and the number of institutions that can do clinical trials. We also don't have the capacity to do phase 3 clinical trials in this country because, fortunately, we don't have the prevalence of SARS-CoV-2 infections in this country that they do in other parts of the world. So that's part of the reason why some of our products may not have got through the phase 3 clinical trials yet.
Okay, thank you. Oop. Hold on. Okay, starting from the bottom. It would be very interesting to get your perspective on nasal spray vaccine.
Okay, something like flu mist, I believe you're sort of relating to. That is the flu vaccine that's given by intranasal spraying to children. I think if you're actually trying to induce an immune response, which is localized, that is, in the nasopharyngeal area or lower, giving it through a nasal misting approach may actually be a wise approach. But it all depends. I don't know any results with that type of approach using a COVID candidate at this point. However, there may be some out there. To be honest with you, there's so many papers being generated right now, I can't keep up with it.
Yes.
So you do the searching, check for good bona fide science results. Don't depend on Facebook or certain parts of the internet.
Yes, indeed. So related to that, Rodney is wondering, he heard on a science podcast that an injected vaccine may provide good immunity to areas with good blood flow and lesser immunity elsewhere. And kind of riffing on that, if you were saying, the localized injection into the nasal cavity might actually be beneficial. So in this case, it's more related to blood flow. So he's wondering if a vaccinated person could have immunity to severe infection, but could they still be infectious? For example, he says he's heard of people who've recovered from Ebola might still have access of active virus in their eyeballs. Comments?
Mm-hmm.
That's a curious one.
I mean, they probably have active virus in several positions. Obviously, you look to see, are they still secreting virus? Is it still infectious? And now, to be honest, they don't do that on most people. And the longer we go, we won't be testing everyone that actually is cured or recovered from this to determine whether they're still secreting virus. The sort of understanding now is that once you're actually virus negative for 2 tests, then you're negative. But it may actually pop up again. We don't know. Some of the early results on reinfection, some of the interpretations was there was just reactivation of virus that was there in some sequestered site and came up again. But the idea of actually vaccinating and for example, your arm versus getting it intranasally, there is sort of a lot of results that would say that your local immune response, your lymph node, for example, close to the point of injection would have the better immune response. But that is cells that have accumulated in the lymph node, but do circulate elsewhere. So, I've seen a lot of results, a lot of experiments that would say actually trying to induce better mucosal immune responses, intranasally would be an example, would be something you want to do. However, we're still early days here in regards to what is a protective immune response and what actually may not be a protective immune response and could actually potentially exacerbate an infection with the virus afterwards. So there's a lot of work that has to be done still on the immune responses and what can be done to dampen a very vigorous pathogenic response.
Yeah, on that note, I've been hearing that the concept of the reinfection, it's that some might have mounted an immune response, but they still didn't get the disease, right? And that another reoccurrence could actually be the disease, or vice versa, right? So some folks may get really sick at the beginning and then a reoccurrence, not more mild, or vice versa. So, I'm just wondering if, yeah.
I think the results I've seen on these reinfections, it appears like for the most part, it's people that have had an initial very asymptomatic mild infection that seem to have come across with a second viral infection afterwards. And that may be because their immune response, or the immune response that they actually induced, was a fairly weak immune response. Mm-hmm. Maybe there wasn't a lot of neutralizing antibody there, if that's actually correlating to protection. Or other antibody responses. I've seen fairly weak antibody responses in some of those individuals.
And I've also heard the criticism that if reinfection was so common, you would expect more, given the population, the sheer number of individuals that have since been or have been confirmed positive. So there's clearly something else, there's more we need to know, obviously.
Exactly. And I think by using vaccination, we actually may be able to induce better immune responses that may put into that protective mode where they have the right immune responses and not the wrong immune responses. And as we know, COVID is part of it is a very active immune response, a inflammatory immune response, which is very pathogenic. You don't want to induce that type of response in your vaccine. So, you gotta be careful about what you're doing. And I know that, especially some of the first groups that were working on this were very aware of that sort of what they call antibody-dependent enhancement of infection. They're very aware of it, and they were working on that as well.
So back to our attendees, Harish is wondering, can bacteria such as Streptococcus that are naturally present in oral cavity act as vectors for the SARS-CoV-2 virus in addition to saliva aerosols?
Interesting. I don't know if it's being used as a viral vector right now. Some of the problems of using bacterial vectors is that they don't use the same type of glycosylation. So you would
They don't use
actually
the same what? Say that again.
glycosylation. So the sugars that are put on, so for example, the spike protein, which is a glycoprotein, so it's a sugar-coated protein, and maybe that sugar coating is important to the type of immune response you're getting, a protective immune response. So if you express in bacteria, you won't get the same type of sugar coating. It's a very different type of response than you would have from expressing in human cells, for example.
Interesting. So much I don't know. This is why I'm here.
Well, yeah. I mean, we were working on some of that stuff back in the late 1980s and onwards. I'm showing my age now. Yeah.
So we've got a couple questions that relate to what's happening in Russia. Olga's wanting to know, could you please comment on the discussion that that came in the Lancet after the publication of the Russian scientists' results?
To be honest with you, I mean, that's getting into science politics. And, you know, I would like to have obviously seen a more fulsome analysis of all the results that they actually generated to assess actually how effective and how safe that vaccine was. A little more information actually on the vaccine, how it was generated, where it was generated, et cetera, would have been very nice. But, you know, ultimately, if they have a vaccine that's effective for their country, then they've done something right. And time will tell. It's sort of interesting. I heard that, you know, Putin injected his daughter, which was sort of, I guess it's the one he didn't like.
Oh my. So somebody else who's anonymous is wondering, based on Russia's approach on skipping phase 3 trials, are there any pipeline steps that can be rushed through or even skipped in order to get the COVID vaccine out in a record time? The timing is insane, and there must be some steps being sacrificed.
Okay. Yeah, it is. I mean, their approach was very interesting. Now, saying, you know, the question is a very good question. And what has happened with COVID is actually they're doing the clinical trials simultaneously. So there's phase 1, 2 that are being done, and there's phase 3 is being done. So they're actually rolling them on fairly quickly, which is very interesting because usually it's sequential. You'll do the phase 1, and then you'll do the phase 2, and then you'll go to phase 3. So this is sort of broken the classical approach of getting a vaccine through to licensure. And you're seeing, you know, like even Health Canada is sort of saying, you know, we'll look at data before the phase 3 is finished. Very interesting. It's absolutely fascinating to watch what's happening. And it encourages me actually incredibly to think that we actually could have a vaccine maybe this time next year.
Yeah, and I'm wondering, I know this is not necessarily your area, but if you could comment related to that on if you think that they'll have guidelines around populations who might receive the vaccine first, for example, frontline, you know, medical physicians, nurses, etc., populations 65 and up, immunocompromised. Do you think that we're going to have?
I'll defer that question. I used to work with the
Okay.
Public Health Agency in the Center for Immunization and Respiratory Infectious Diseases, and they support a group called the National Advisory Committee on Immunization. And they would be making recommendations on COVID. In fact, I know there's a working group over there, but I'm not involved with it.
Right, but so, yeah, I guess I'm just wondering, there is clearly conversation on this, and there probably will
There is
be some
definitely,
recommendations.
but I'm not privy to it.
Sure, no worries. All right, next question from Olga wants to know, does anyone use AI or simulation? Yeah, like computer.
Very interesting question.
Computer models, yeah.
Yes, very interesting question. There's a group actually out of Cambridge University, and a Canadian actually that's involved in it. Jonathan Heaney, a veterinarian who trained at Gulf.
Cool.
He's been working over in Europe for I don't know how many decades now, but he actually has an approach, and they were using AI as an approach to try to look at all the different types of coronaviruses that are out there and try to get a common sequence so you could actually induce an immune response that would protect not only against SARS-CoV-2, but maybe things that would come out after that, because this may not be the only coronavirus that comes at us in the future.
Yeah,
so and yes, they definitely were using AI to
that's really cool.
Generate that product.
Because I've been wondering too if, based on this rapid R&D, this rapid, you know, like global response in creating the vaccine, if any of it's going to be translated to future diseases or future vaccines, you know, for example, for various flus,
Yes.
right? Can we harness what we've learned through this global response for the production and manufacturing of future vaccines?
It may actually help in regards to, you know, some of the vaccine, flu, for example, it takes up to 9 months to actually get it into our arms. They'll make the call on the vaccine in February, and I got my vaccine last week. So there you go, 9 months. And I was lucky, I went to Costco right away as soon as I found out. But yeah, it may actually reduce that timeline because maybe they'll be able to do the clinical trials more simultaneously and get it out quicker. There's other companies out there, Medicago, for example, that's generating products in plants. Their advertisement pretty much is that they can reduce that timeline down to maybe 3 months versus 9 months. Wow. Exactly. So then all of a sudden you're vaccinating kids before they ever get back to school.
Wow.
Yeah, so yeah, I mean, this is sort of where the field has been evolving over, you know, a couple of decades now, but I think this is going to help. Now, the other interesting thing that's happened here is that the initial discovery of this virus was fairly rapid because there was a lot of international work going on through sort of a global effort to try to look at the virome, what actually is out there circulating. And it's amazing the number of potential viruses that could come at us. So it was sort of interesting that this group, especially the group in Wuhan, was very involved with the Americans and looking at what potentially is out there. And that could potentially help us as well to generate vaccine candidates that may come towards us.
Yeah, so related to this, we have two questions. These are wonderful questions from our attendees. So Catherine's wondering if you can comment on the mutation rate of SARS-CoV-2 and how that might impact vaccine development. And then we have an anonymous attendee that has added something, or another question is related, so I'll put them together. Given the mutations in SARS-CoV-2, do you think new vaccines will be needed to be developed every year for COVID-19, or would booster shots be sufficient? So kind of, if you could comment on that, sort of the rapid mutation rate and what that might mean for vaccine development.
Exactly. Now, this is an RNA virus, no question about it, but it's a very complex RNA virus. RNA viruses traditionally have very high mutation rates. Flu, for example, very high mutation rates. That's why we need new vaccines every year. Now, coronaviruses, by their nature, actually have an error recognition and repair mechanism. So their mutation rate is probably about, I've heard, about half of what flu would be. So yeah, there's some mutations out there, but we don't even talk about these as strains. They're variants. They're not even out to the strain level.
Unlike a flu,
so
which
would it be a boost? I don't know, maybe.
Yeah.
But I would say that, you know, we're already sort of seeing that maybe we're going to have to boost anyways. So if you're going to boost anyways, then look to see what's circulating at this time point and make sure that the vaccine you have actually will work against that variant is probably what's going to be done. If it isn't, then they'll generate a product accordingly. But they'll have the vaccine approaches, especially if it's the RNA approach or some of the other approaches, where they can take one out and put another one in and fairly rapidly run the clinical trials to do that. So it is a very different story than it might have been 20 years ago, for example.
Very cool. Thank you. And then is Carleton doing or able to do any research on COVID-19 vaccines?
Well, to work on live virus, I believe you need a level 3 facility, and I don't think we have a level 3 facility at Carleton. However, there is facilities at the University of Ottawa and elsewhere in the country, so work can be done. If you want to do work in preclinical testing, for example, that can be done out in Winnipeg, and I believe Vito Interfac in Saskatoon has that capacity too. So work in collaboration. This is very much about collaboration. To be honest with you, most of my vaccine work was never done in one institution. The ideas were generated in one. We tried things, preclinical testing in another one. We did, you know, work with the people on scale up in another institution. That's the way the work is done. It's not done any single location anymore, well, at least not often.
Yes, this is definitely a collaborative response.
And, you'll see most of my papers, for example, are many author papers because there's people from several institutions involved. I mean, when I worked in Europe, it was fascinating because the Europeans truly collaborated. So we had concerted actions on HIV vaccine development that were truly international initiatives. And it just made the work so much faster. And it was great actually being able to visit different parts of Europe and meeting people.
Yeah, that's one of the best parts about being a scientist.
Very much so.
Anonymous attendee wants to know, what are examples of adjuvants used in vaccines, the vaccine against SARS-CoV-2 or others?
Okay, there's several. There's, I mean, alum has been used in the past. So this is an aluminum hydroxide, and it was implicated in this connection with autism, which was debunked after spending millions of dollars to debunk it. And the individual, Wakefield, actually, who generated this, was debunked as well, unfortunately. So there is that product. There's another adjuvant called M. I'm not absolutely sure what that product is. There's a lot of sort of intellectual property on adjuvants, so you don't necessarily know exactly what they're using.
Like thimerosal, methyl mercury.
Those are more
preservatives, or
preservatives. Yeah, it's the alum that was the adjuvant in that case. There's others that we use experimentally, but you wouldn't use it in humans. Freud's complete adjuvant, for example, which is great for immune responses, but you'd never put that into a person. Now, there's some, Freud's complete adjuvant, BCG. We're hearing about BCG as a potential vaccine approach, where you can give people the vaccine and induce a generalized immune response that may protect you against multiple pathogens, maybe COVID as well. So there are some clinical trials looking at that right now. And that would, that's sort of interesting. BCG has somewhat an adjuvant activity. It's one of the components of Freud's complete adjuvant, at least part of it.
Very cool. So somebody else, anonymous, is wondering that they've been advised that various vaccines' effectiveness may vary in different groups of the population. Given this, what are your views for seniors as to waiting until more information concerning the effectiveness is available? And part B, having a second vaccine if a more effective one becomes available, where their compatibility be dependent on the different vaccines. Thank you for your presentation.
Yeah, excellent. Thank you very much. And excellent question. Interestingly, one of the first projects I worked on in virology in 1978 was, why did the elderly not respond well to the flu vaccine? And we're still,
what was the reason?
We know they don't.
Still don't.
And the vaccine that's been brought out to help that is to boost the amount of protein. So the dose is higher in the vaccine used for the elderly, for the over 65s. In the flu vaccine case, for example, unfortunately, the one that my wife was offered, only it was the trivalent vaccine, not the quadrivalent. Trivalent vaccine only has 3 potential flus that would protect against, not 4. Whereas the one for the under 65s has 4. That's quadrivalent. So interesting. So yes, I would say that we already have an idea that the elderly do respond differently. They have a fairly inflammatory immune response. We'll have to be concerned about that in regards to what type of immune responses you're inducing. In the young, likewise, different immune responses, very, very young. And then, the middle between 10 and 65, another group. So yeah, I think it has to be tailored for each one of them. In different groups, maybe different responses. So yeah, the clinical trials will have to be tried. Things will have to be tried in different communities to see how much protection can be induced.
Protection, or even, like you say, the opposite, like preventing the cytokine storm. Is this what we're talking about with that? Like the immune response being so huge, right? Which apparently kids don't have that. They're not, and this is potentially why they're protected from some of the ill effects of COVID. It's because they don't mount this massive cytokine response.
Exactly.
Okay, we're down to a couple more questions. Well, one, Harish is thanking us both. Thank you for the interesting and very informative session. So thank you. And then Rodney is wondering, do annual flu vaccines go through phase 1, 2, 3 trials?
They have in the past. So, I mean, any of the flu vaccines, for example. However, they're pre-existing, so they usually just do the scale up because they've already been tried. So all they're doing is doing a tweaking of the existing one. So that's very different and totally new and novel, as is COVID.
Right.
Yeah,
that makes sense. So panel, I keep saying panelists, attendees, please continue to ask questions. I believe we have more time. What time are we meant to end at? I've got 3 on my schedule, but I don't know if that was for me specifically. 2:30. Oh, look, we're out of time. Well, that was perfect. Perfect timing. I was going to ask about what your thoughts were on. They say now that wearing a mask actually is doing something interesting in that if you are exposed, say, to like, the aerosolization of COVID, SARS-CoV-2, that you're getting like these little mild amount of the virus breathed in, and that might be actually providing some small degree of immunity. So I'm just wondering if that's what have you heard that?
I mean, it's interesting that there's several studies where we look at low dose immunizations versus higher dose immunizations. Some of the attenuated HIV vaccine approaches using the monkey model that I was working on, you will see different responses depending on how much, what dose you actually use to immunize. Right. So yeah, maybe inducing is very, very low level, but
very minor, and then
yeah. To be honest with you, I don't know. I mean, what is the dose that actually causes an infection with COVID? I'm not absolutely sure of that. If it's, if it's 100, then getting 20, maybe you won't be infected, but you'll actually bind them into your mucosal. Maybe you'll induce an immune response, but it's just so low dose that it's not actually starting a proper infection. Maybe. But to be honest with you, I still wear a N95 mask, or we actually make our own, which are 3 layers. We actually have a flannel layer in between, and it's pretty much, if I'm thinking of how hard it is to breathe, it's pretty much the level of a N95 mask. So I'm not taking a chance on that one.
Fair enough. Olga is thanking us. Thank you. And so I do want to close by also acknowledging this was a wonderful conversation, and I'm grateful to have listened in and to moderate this conversation. Lots of great questions from our attendees. So once again, thank you, Dr. Rudd. I hope you can hear us all clapping.
Thank you.
But we do so appreciate the time.
And I'm going to say one thing to you, Kim. Congratulations on your award. I understand that you won an amazing award recently.
Thank you. Yes.
For your teaching, so I am inspired.
Yeah. Yes. Well, we have a great community here at Carleton, so lots of things to celebrate. So thanks everybody for joining us, and stay tuned for further science cafes. Have a good rest of your day. Bye.